Okay, then we will have a much relaxed session, also the last session of the afternoon. So basically, just give you some demonstration of the package. Just remind you that we have actually two types of search. One is a dynamic neighborhood search for the most for the best enrichment of the input gene site and in the network. And then we have the static neighborhood search. It is quite straightforward. So we just look at the downstream and the immediate R degree. And then we rank all those enrichment types all degrees and nominate global driver and local driver. Okay, so yeah, Janet already uploaded packages, one for Windows and another one for Linux to the course website. But when I get back to Mount Sinai, then I will, because we will keep actually improving the packages. So this should be the permanent location for holding the package. And I will keep you guys posted on the release of the next version. So the installation is quite simple. I think since many of you are very familiar with R. So in this case, I install in a local directory. And to test, then you just, there are two demos, and one called like breast cancers. That is the case for the static neighborhood search. So the input is the nodes of the whole network, and you just compute it downstream and also the immediate R degree to figure out the top drivers, the drivers. And also the demo of east, actually, that is an example for the dynamic neighborhood search. Okay, so we can try it now. Okay. Yeah, the east example will take quite some time because we have 13 actually equidal hotspots, and then we need to search all of them. So the breast cancer actually example includes 2 modules actually. Okay, so this actually is part of the work actually that is still in review, but I would like to share with you guys now. So that is includes the 2 modules we identified through the WGCNA. So those modules are highly predictive of clinical outcome, the survival. So when you run this demo, yeah, so you can see, so it is really, yeah, this actually, okay, yeah, I think the problem is that we, yeah, okay, run again. Yeah, so basically, actually, because the input is what? Okay. Yeah, because we use this kind of static neighborhood search. So basically, we just compute it downstream. So as you can see from the report of the screen, then you can see that the top driver is like aura kinase, and yeah, so now actually it goes through. There's many warnings actually, but it's not important. So this is the ranking order of the drivers. So you have a lot of information about the downstream size and also the network size and also the layers we searched. Then we have the a variable like a key driver to tell you whether it is a global driver or local driver. One means like a global driver, and zero means like a local driver. Because it is a static neighborhood search, actually, those p-values for the changes actually are not meaningful. Let's see. Okay. Okay, so the input actually is very simple. So you basically take a list of genes, but in this case, actually, I make it a little bit complex, but for each, but make it the user actually more friendly, because you may have like a multiple lists. So what you can do is just let me check. Yeah, it's just like as the output of WGCNA, you have the props and the module membership. So that's I just require you that the first column is the node name and the last column is the membership name. And then for the course network, just basically two columns. The first column is the course for the second column. You can put additional information like probability of the causality between the two genes. That's it. And then this method can also work on directed or undirected network. So you just specify whether the network is directed or not. And then you can make a choice of the number of layers you want to search. And yeah. Yeah, and yeah. So since the input is a list of like the module genes, and then we actually identify drivers for each of the module. Okay. So we have some the default settings for those parameters, and so the important thing is about the output. So the output actually is actually a number of files. So you can see, let's go back to, yeah, so you can see those breast cancer samples you can see here. So this is the blue module. So you have the CYAs, that is abbreviation of a Setscape. So you can, this is the network file. So you can see like this is the source node. Those are the destination nodes. So that means actually the first node is the causal for the second one. So this is the subnetwork, is a module specific subnetwork. In this case is a blue module. And then you have the nodes. So you have the network. Then the nodes actually tell you, yeah, let me see. The node actually properties actually were set up so that you can directly input into Setscape and for a quick visualization. So you can see here like for each node you have different colors and all the shape and the size. So obviously for the big nodes, they are, and those bright colors, and they represent those top drivers. And you also have the font size to give you a better visualization of the node names. So in this actually SNP file give you some like annotation of the shapes and the color and the sizes. And so I give you a short yeah, so here yeah, so this SNP file tells you the what does those shape and the color and the size actually mean. So in this case, you can see like SIG, that is that means actually this gene is in the input gene list. So NCIG, SIG means actually this particular node is not in the gene list, because especially for those dynamic neighborhood search, so you don't expect those driver genes always from your list. They may be from outside the list. And so we have this kind of notation, and GKD is a global key driver, and LKD is a local key driver, and NKD is a local driver. So we have this kind of network, and so basically eventually you just need 2 files from the output. So you can see this kind of CYS file, that's the network file. And then you have the node property file. So we have a Cytoscape plugin to visualize, but last night I couldn't upload it to my home page, but I will update it later. Let's see. Yeah, we have a plugin called like Sage plugin. So I will upload it on the website, so you can directly input the output from the key driver analysis, and so you can see. Let's see those blue module, and also there's the node. That's it, and then you can see those red nodes and the blue nodes and the drivers, and then you can play with the Cytoscape for different layout. Yeah, so that's very actually straightforward, and so the key thing actually is to get the right gene list and the right course network. So that's all actually we talk about today, like the structure way to build up network, and the genetic-based network all can be used as the input for the KDA. Yeah. So yeah, I think this is a very short demo.